So next we have Dr. Isaac Sandoval, who is an assistant professor in biology at Dartmouth University. Dr. Sandoval received his MS and PhD in chemical and biological sciences at the National Polytechnic Institute in Mexico City. And just a note, I earlier said Dartmouth, I meant to say Georgetown University. He focused on physiopathology and host immune response during his graduate work and has now moved into a more challenging field, neuroscience. So Dr. Sandoval will be discussing the science behind memory forgetting. And without further ado, please welcome Dr. Sandoval.
Hi, everybody. Can you hear me well? Can you see my screen?
Yep.
Okay, thank you. Well, first, I want to thank Diana for this wonderful invitation. I think I always have thought that it's very important to communicate science to young guys, to communicate the excitement that science produces. Ever since I was a kid, I knew I wanted to be a scientist. I don't know how that happened in me, but I never had doubts about what I wanted to do. And in biology in particular, I was always very interested in knowing how live things work. I remember when I was a kid spending a lot of time looking at animals or insects and just wonder what was going inside their bodies to behave like they behave. For me, they were like small machines, and it was just so mysterious to know like how what's going on inside their bodies that makes them alive. So becoming a scientist was not a question for me. And then I just want to convey you to tell you that there's nothing more enjoyable than finding something that nobody knows. Like when you are in science and you were conducting an experiment and all of a sudden you have an epiphany or you find something that is completely new and nobody knows about it. It's just the amount of energy that you get from that is just amazing. It's one of the best experiences you can have. And so that's why I became a scientist. And I started my career, I am from Mexico, and I started my career working on a parasite that infects the brain, especially of kids. And it's a very dangerous disease. But then I remember when I was in college around the year 2000, I was reading about this gastropod in the ocean called Oplecia. And I was reading that a guy got a Nobel Prize because he discovered how, or the molecules that are involved in how the memories are encoded in the brain. His name was, Eric Campbell. This is his picture, and this is the Aplysia. This is a mollusk that lives in the ocean. And using this animal, he started to discover how the memories are encoded in the brain. And I was in college when I read about him, and it was just fascinating for me. And so that's why I decided to go into the field of neuroscience, and particularly in learning and memory, to try to understand how our memories are encoded in our brains. I have to say that I got to meet Dr. Kendall a few years ago when he visited the institute where I was working in Florida. And so this is some of the things that are also very exciting when you get to know the people that inspire you to in your career. And he was one of my heroes. I have many, but this guy was the one that made me decide to go into learning and memory field. And so my lab now is trying to understand exactly that, how our memories are encoded in the brain, how the memories are consolidated, meaning that how these memories that at the beginning, when you learn something, this memory is very labile. It can be forgotten very easily. But some memories, if they are really relevant to your life, then they become what we know as long-term memories, memories that can last for a very, very long time. And that process is called memory consolidation. So my lab started to work on how memories are encoded, how memories are consolidated. But very recently, we are also very interested in trying to understand why do we forget? and how do we forget? What are the molecular mechanisms behind that memory forgetting? And so usually when we think about forgetting, we think that forgetting is something bad, right? Like because we associate forgetting with some very devastating diseases like Alzheimer or dementia, and also forgetting something important can get you into real trouble. I remember one time when I was in college, actually I was in my PhD, and they invited me to give a talk on whatever my research I was doing at that time in a city that it was far from Mexico City. So this city was like 4 hours far from Mexico City, and I remember this talk was in a very important museum, and it was a big deal. And so I decided to use a nice jacket and a shirt and a tie. So I remember I traveled the day before my talk, and so I packed everything in my suitcase. But I decided to put my jacket, my shirt, and my tie in a different bag, a bag special for that. And so I traveled the day before, and this talk was in the morning. And I remember I woke up with enough time. I took a shower, and then when I was about to dress up, I realized that I forgot my jacket and my t-shirt and everything, my shirt. And so that was very scary because I didn't have any other clothes proper for this talk. So I went out in the city and tried to uh, look for a store where I could buy something, but everything was closed. It was really, really early. So I was just like knocking in doors of the stores, but hoping that somebody was inside. And yes, I found somebody that they were about to open the store. So they opened it for me and I got to buy something, and it didn't fit well, but it was enough. So yeah, I mean, forgetting is if you forget something important can get you into real trouble. And I'm a very forgetful person. This has happened to me many times. And so when many times I found myself thinking, it would be wonderful if I don't forget anything, if I could have a perfect memory. But then when you think deeper about this, you start to realize that forgetting maybe is not such a bad idea. And this is very well represented by a fictional character called Funes. And this is a drawing of him. This is a fictional character of a book from the Argentinian writer Jorge Luis Borges. And this guy was a guy that he could not forget anything. For him, his memories were perfect representations of the past, something like a videotape. And so he could remember all the details about everything that he experienced. And that was okay. I mean, when somebody told you that, it sounds like could be fun. Like you can remember everything. But he also had a bunch of cognitive problems. Like for example, when he was trying to remember one day of his life, it would just take him one full day to remember a day, right? Because he remembered every specific detail. And so he could not live in the present. He was constantly living in the past because also every time he saw something that triggered memories in his mind, he was just going back to the past, and he was not able to live in the present, along with many other problems. And so when we think about, intuitively, we think about what forgetting is doing for us. Why is important that we forget? We came out with at least four possible causes. So one is that if you think that your brain has a limited capacity to store information like the memory of your computer, and you experience all these things every day, the colors in the sky, the people walking, the things you read, the things that you see on TV, all this information, if it will be stored in your brain, it's so much information that probably it's going to saturate your brain pretty fast, and it will leave you very little free space. But that doesn't seem to be the case. We always have more capacity to learn new things, and we think that forgetting is really important for that. So getting rid of things that are not important anymore, that you don't need, and then you just can just get rid of them. And so it's important for getting for that, so to be able to have free space in your brain. Another cause is we think that forgetting is really important to increase efficiency during memory recall. And I'd like to give this example. Let's say that this desktop of this computer is your memory system. And all these files here are the things that you remember. Now, let's say for a moment that you need a file from all this mess, and you need a picture or something from this desktop. And then if you try to look through all this mess, it's going to take you some time, and it's going to be very slow to find the piece of information that you need from all this mess. On the other hand, if you keep your system clean by means of forgetting, so again, getting rid of all the information that you don't need anymore, then your system is going to look more like this. And it's going to be way easier and faster to find what is important for you at the moment. And so forgetting is really important for this efficiency during memory recall. Another thing is that we live in a very changing environment, right? So our memory, our environment is changing all the time. Things are changing all the time. And so forgetting is really important to maintain what we call behavioral flexibility. So you need to be able to update your memory system to forget things that are not important anymore and to update it to new situations. For example, let's say you live here and you have a favorite restaurant and you know exactly where it is, you know exactly what path to take to get there. But let's say they move this restaurant and then now you need to update your memory system. Now you need to update the new location and to learn where is the new location of that restaurant. And with time, you're gonna forget about the old location. And so memory forgetting is important for this kind of updating, memory updating. And lastly, forgetting is really important for promote generalization. And I'm gonna explain you what this is. Let's imagine that you are a mouse or a rat or something like that, and then you have encountered several cats in your life, and now you know they're dangerous because they chase you, they try to eat you. And let's say these different cats have different colors, different sizes, different size of fur, different odors. And if your memory system was a perfect representation of each one of these cats, then your memory system will look like this line, okay? So let's say these blue dots are each one of these cats, and their position in this graph is given by their different characteristics. And so if your memory is perfect, then your memory is going to look, your memory to predict what a cat is going to look like this black line, okay? Now let's say you find a different feline, like this is a bobcat, okay? Which is completely, not completely, but somehow different. It's bigger, it has different scents, different color, and that's represented by this green dot here. Now, if your memory is perfect, then this memory is going to fail to predict that this is also a feline and this is also dangerous. And so this can be very bad for you. On the other hand, if by means of generalization you forget about all the small details of every specific, but you just keep the gist of the information of what a cat is, what a feline is, then your model to predict what a feline is going to look more like this black line. And then this model is going to be better to predict that this bobcat is also a danger for you. And so this process is called memory generalization, and forgetting is really important to achieve this. Okay, so in my lab, we are trying to understand how do we forget? What are the genes or the proteins that are involved in this process of memory forgetting? And to do that, we use the fruit fly. This is the animal model that we use. This is called Drosophila melanogaster. And we use this animal because it has a lot of advantages. So it's really small. It has a great amount of genetic tools. People have used this animal for over 100 years. So they have developed a lot of tools that allow us to uh, dissect their brain in different ways, to mess up with their brains, to control their brains. And so it's a very, very useful model. Also because it's so small and they can live in with very little, it's a very cheap model to work. It's low cost, and also in time we can have as many flights as we want in around 10 days. That's the time of the life cycle of a fly, compared to, for example, animal models like mammals, like mouse or rats, where you have to wait for months to get animals and start working with them. and you cannot have as many, and they're expensive. So Drosophila is a great model for that. So how we do this? Well, the first thing we do is we train the flies. Okay, so we teach them stuff, and yes, the fly can learn. What we do is we put small flies in a classroom, and when we lecture them about the dangers of electricity, and they learn. I hope somebody's laughing because that was a joke, but we do something similar. Okay, so we what we do is we put flies in a tube, an empty tube that looks like this. We put a bunch of flies there, and then we could pass an odorant through that tube, and so they can perceive that odorant. And then at the same time, we give them like mild shocks to their legs. And so normally the protocol works like this. So we put the flies in at the present of another one for around 1 minute, and then at the same time, we give them 12 shocks of 90 volts in their legs. And so they associate the odor with something bad, with the danger. And they learn this association. We then let them rest for 30 seconds, and then we present a second odor for 1 more minute. And this odor is not paired with anything. So it's just odor. After this learning session, we can put the flies in a T-maze that look like this, and we can present other one and other two in two different arms. And then if the flies remember that association between the odor and the electric shock, they are going to avoid it. So they are going to run away from there and that and they are going to position themselves to the other arm with the other odor. And so in this way, we can calculate the memory. We can estimate how much they remember from that task. This is one of the things that we do. So we do behavior. We analyze the behavior in this way, how much they remember. But we can also look into their brain. So we have these powerful microscopes where we can put a fly under the microscope, and then using some genetic tools and some fluorescent proteins, we can actually record the brain activity of a fly. And so what you see here is a fly trying to walk around. It's fixed in a chamber that is below this microscope, and then it's moving, it's alive, it's trying to walk, trying to fly. And at the same time, we are recording this video that you can see here in the upper panel is the brain activity. So every time you see these flashes of color, that means that the brain is very active. In this case, we are recording from this area of the brain, which I'll present you a little later. And so in this way, we can also, train the fly under the microscope. We can put an electric reed touching their legs, and we can deliver odors. And so we can train the fly while the fly is learning, and then look at the brain and see what is going on in the brain as they learn or as they forget. We do also a lot of microscopy, so we can look with a super resolution microscopy. They're part of the brain that is important for memory encoding. In this case, we are looking at this region here called V1, and this is a lot of detail of all the synaptic connections, so connection between different neurons and how they change with learning and how they change with forgetting too. This is the Drosophila head, and this is their brain. This is how it looks. These two lobes that you see here, they're the optical lobes. They're the ones that receive the information from the light. And then these green things that you see here, they're called olfactory bulbs. And they receive the olfactory information. So their fly noses this thing that you see here. This is the antenna. And so others are perceived with this antenna. And that information is sent to the brain, to this area. And that information then is conveyed to the back of the brain and gets to these L-shaped structures here called Martian bodies. And this is exactly where the memories are encoded. These are the sites of the brain where the memories get stored. And also, of course, is the place of the brain where the memories are forgotten. Okay, so this is a kind of a complicated model of how we think memories are encoded in the brain. Normally, the cells or the neurons that perceive the odors are called canyon cells. So these are represented by these green circles here. And then once they receive the information, that information is going to go down and is going to be conveyed to two different kind of neurons, these blue ones here and these pink ones here. And these neurons are going to drive the approach behavior. So when you activate these neurons, the flies like it, and so they will approach to whatever stimuli that they are sensing is rewarding. And on the other hand, we have another branch, another kind of neurons that when these neurons are activated, they avoid whatever is causing this activation. They don't like it. So that is an aversive stimuli. And so normally when we have another that is neutral, that it doesn't cause any behavior, the flies don't care if they don't know if it's good or it's bad. One of these cells is going to be activated, and then it's going to activate these two branches in a very equilibrated way. So at the end, the result is nothing because you have the same amount of approach, the same amount of avoidance, and so the result is no behavior, nothing happens. Now, during aversive learning, what happens is that you have the activation here of a specific odor you're using for training the fly. And then at the same time, you have the aversive stimuli, right? So here you have the electric shock that is conveyed by these neurons called dopaminergic neurons. So these are called dopamine neurons that produce dopamine. And then when you have both the stimuli presented at the same time, you have a point in this circuit where there is a coincidence. And that point is going to be depressed, basically. And so now what is happening is that the next time that the fly experience that same odor, that information is going to go down again. But since this node or this point is now depressed, that is going to result in less activation of this branch. And then the result is a reduced approach. And since the other arm of this pathway is still normal, and then now the avoidance has a bit higher weight, right? So the balance between a reduced approach and a normal avoidance is avoidance. And so now, after aversive training, the fly is going to avoid that particular odor. And so that's what's happening in the brain. And the same thing or a similar thing happens with appetitive training. So we can also train the flies to associate odors with a reward. In this case, we feed them with sugar, and very similar, something happened in that case. Now the avoidance branch of the circuit is reduced and the approach is still normal. And so the end result is the flies like this odor and now they approach it. Now, when we train the flies, we can score that memory and we call that performance index. So what this means is if we have a score of 1, that means that all the flies remember the task. So all the flies avoided the odor that was paired with the shock. And if the score is 0, that means that they don't remember nothing. And so when we train the flies and we test their memory right away, most of the flies remember the task really well. But if you test the memory like sometime after training, they start to forget, just like we do. And if you test the memory around 24 hours later, most of the flies forgot the task by that time. Now, just like in humans, I know you experience this, right? So when you, let's say you have a test and you study once, only once, and you have this test right away, maybe you can do okay with one test, one training session or one study session. But if you wait long enough, at some point you're not going to remember anything. But let's say instead of studying only once, you study many times, and you study, let's say now, and then you have a rest, and then you study again, and then again, and again, and you study many times. Then this memory is going to last for a long time. And the same thing happens with flies. If we train the flies 5 times, they will remember for a long time. This memory can last many, many days, even like 20 days or 30 days. And taking in consideration that the life of a fly is around 60 or 50 to 60 days, that means that this memory can last at half of the time of their life. It's a very long-term memory. And this kind of memory requires protein synthesis. So that means that new proteins have to be synthesized in order to form this long-lasting memory. Now, so when we wanted to understand what are the genes or what are the molecules that are involved in this forgetting process, we reason, okay, let's do something. We can manipulate genes in flies very easily. And so what we do is like, okay, let's mess up with the function of a gene and see how that affects their memory, and see if that affects how they retain their memory. If maybe we can have a fly that remember for a long time. And so we did that. We messed up with the function of around 3,000 genes. That took around 3 years and the work of 4 or 5 people. And at the end, we found a series of genes that look like this. And this is one of them. This is called Scribble. This is the name of the gene. And this is a 3-hour memory score. So that means this point here, when we train the fly, we wait 3 hours and test the memory. And these are control flies when we don't do anything with the gene. And this is when we interfere with the function of the gene. Okay? And so you can see how these flies actually remember better. Now, this gene was discovered in the year 2000 and by a professor called Builder. And he found that this gene was expressed in the epithelial cells, basically. So in the cells of your skin, in the cells of your gut. And this protein was really important for what is called cell polarity. So if you take cells from your gut or cells from your skin, and you will notice that the top part or the apical part, the top part of your cells is very, very different from the bottom part of your cells. And Scribble, this protein called Scribble, is really important to make that difference. And that difference is fundamental for the function of your cells. So if you don't have this, then you cannot survive. And Scribble is really important for that. And then we found that this gene, it was also expressed in the brain, and it was important for memory forgetting. So when we knock down that gene, or when we interfere with the function of this gene, this is what we see. This is the memory curve that we found. So no, this black line is the memory decay of a control flies, of control flies, and this is the memory decay of flies where we basically interfere with the function of this gene in the brain. And you can see how these flies uh, uh, remember way better. Do we still have time? Sorry. Diana, how much time do we have? Sorry.
Yeah, we have, right now we're approaching the 30-minute mark, so we have a few more minutes.
Okay, sorry, I'm going very slow, but yes. So this is what we found. Messing up with this gene makes the fly to remember better. And then if we look and look at the memory of these flies for a very long time, these flies remember this memory for almost 60 days later. So they basically never forget about this. These are super smart flies. So we reasoned that there could be at least 3 reasons why this is happening. One, maybe the flies just learn better. They are super smart. They study one time, or we teach them one time, and that's enough for them to form a super robust memory. So memory acquisition, so they are just better at acquiring the memory. It could be that they have enhanced consolidation, so that means they are better at taking a label memory and transforming that memory into a long-term memory, a protein synthesis dependent long-term memory. Or it could be that these flies are impaired in memory forgetting. And so we did several experiments to try to see which one of these three options is what we are seeing in these flies. And the first thing we did is that we trained the flies with a mild training protocol to see if these flies were learning better. Okay, so that happens to you too. If you study just a little bit, you won't learn as well as if you study a lot, right? So here we train the flies instead of using the 90 volts that we normally use. We use 45, 20, or 10 volts, and you can see here the control flies and the scribble flies in red, and you can see there's no difference. So there's very little difference. If any, it looks like the flies are worse to learn. And this is more evident when we change the number of shocks. We normally use 12 shocks, but when we use 1 or 2 shocks, it looks like actually these scribble flies are worse at the acquisition part, worse at learning. So no, it's not this. They don't have an enhanced memory acquisition. Now, we knew that protein synthesis is important for taking a label memory and making a long-term memory. And so we wonder if this is what it was happening with Scribble. And so what we did is we trained the flies, and then we treat them with a drug called cyclohexamide, that basically what it does is blocks protein synthesis. So this drug don't allow the body of the fly to synthesize new proteins. And normally in control flies, that is going to cause a decrease in the long-term memories. But you can see here how the Scribble knockdown flies, there is nothing. Nothing happens with this drug. So that means these flies are not forming protein synthesis dependent long-term memory. It's just a normal memory that lasts a very, very long time, but it's different in nature. And so it's not memory acquisition. And so what it got left is, okay, let's say if they have an impaired forgetting. And what we did here is something very simple. So we trained the flies to associate the other A with the shock, and then we present the other B with no association. And then right after that, we say, hey, no, that was wrong. Now let's learn this new thing. Now the other B is paired with the shock, and the other A is not. So normally what happens in control flies is that this new learning phase is going to interfere and it's going to induce a forgetting of the initial learning. And you can see this by this graph. This is the initial learning, and then after the interfering session, that memory is decreased. So that means the interfering session has induced some forgetting. But that doesn't happen in the Scribble flies. Scribble flies, they just don't forget, they remember forever. Okay, so conclusion, these flies have an impaired memory forgetting. Then we wonder where in the brain is this protein required? And so using all the nice genetic tools that we have in Drosophila, we can actually go and interfere with the function of this protein in cell-specific manner. So we can decide where in the brain or what neurons in the brain we want to interfere with this gene. And so we found that when we interfere with the function of this gene in the dopaminergic neurons or the motion body neurons that called Kenyan cells, we have this enhanced memory. And so then we did some microscopy and we found that actually Scribble seems to be expressed here. And this is what you can see here in green. Okay, so this is the Drosophila brain, and the green part is basically a technique to detect that protein called Scribble. And you can see how it's highly expressed in these L-shaped structures called motion body, which we knew are the parts where the memory is encoded. And so these kind of experiments now allow us to understand better what are the genes and what are the molecules that are important for us to forget memories. And it's not a surprise that this gene was then, a few years later, found that it's important for memory processing in humans too, mammals and in humans. Not only that, we also found that sleeping is really important for memory retention. So in the fly, one of the other tools that we have is that we can install switch in neurons, either light switch or temperature switch, but basically we can install switch in neurons and then we can turn on or off neurons at will. And so we knew, but some other researchers found what neurons in the brain are important for sleep. And so we decided to install a switch like this in the sleep neurons and then turn it on. And basically what you do is you put the flies to sleep. And this is what you can see here. This is a measure of sleep. This is the control, these are the control flies. And then when we turn on that light, that switch, they basically sleep more. And that resulted in a better memory. Okay? So forgetting or memory, like the memories are protected from forgetting when the flies are sleeping. And that's similar, something very similar happens in humans. So this is our final model of how we think things are happening. We found not only Scribble, but we found many other proteins that are important for memory forgetting. And so we believe that really what is happening is that Scribble is somehow like a scaffold. So Scribble is a protein that is binding to many other proteins and is putting together a signaling complex that is conveying the forgetting signal to the canyons, and that neurons, those neurons that are important for memory acquisition. And so we now know all these proteins, and all of these proteins are involved somehow in modifying the skeleton of a cell called cytoskeleton, and that is what is inducing the memory forgetting. And so this is a work that it took from 2010 and up to today, and this is what we have found so far. Okay, I just want finally to thank everybody for listening. These are the members of my lab, some the technicians, undergrad students, and grad students. And I also want to thank my collaborators. And you can visit our website at this link here, cervanteslab.org, and learn more about what we are doing. This is a very nice picture of Georgetown. And if you ever interested in how we forget or why we forget, we can work together. Thank you.
Thank you so much, Dr. Sandoval. We can take like maybe a few questions if you guys have any right now. Oh. Dr. Sandoval, can you see the chat? So
that's what I'm
one question. Okay.
Okay.
Okay, cool.
Can you read it? I don't see.
Yeah. Can we tamper with Scribble KD and human brains so our memory is enhanced for school exams?
Yes. Unfortunately, we cannot do that now. We cannot do experiments with humans. People don't like it. I don't know why. No, well, yeah, we cannot do that. But there's some efforts on trying to develop drugs that will interfere with these genes. So we cannot manipulate the genes like we do in animals, but we can develop drugs that interfere with the function of those genes. And so in a way, try to improve memories, right? For example, especially in some pathological conditions where memories are impaired. In normal humans, like in the normal cognitive functions, I don't know if it's a good idea to mess up with the forgetting part. It could result in some other problems. Like I say, forgetting is normal. Forgetting is something that happens normally. It's something that is important for us to function normally. So you don't want to mess up with that in a normal condition. Okay, I can see the chat now.
There's one. Are there studies being done to explain why we forget some memories more easily than others? Yeah.
Yes, this is a really interesting question, and this is something that we have asked ourselves in the lab. And if you remember that curve of the aversive memory, and when we train the flies with electric shock, they do forget pretty easy, and then more or less 24 hours later, they don't remember anything. But I didn't mention, but if we train the flies with the appetitive conditioning, that means something good with sugar instead of the punishment, they do remember that for a very long time, okay? So that memory lasts way longer. And we are trying to understand why is that? How come some memories that last really easy and some last forever? And in this case, it seems like it is the taste of the sugar combined with the caloric content of the sugar that somehow our brain can detect and basically signals to say, okay, this is really important. Remember this because this is important for you, for your life. This is important for finding food. And so those kinds of memory get encoded in a different way and they last forever. And so there are many ways to approach this question, but basically, if the memory of what we are learning is really relevant for our life, then the chances that it forms a memory that will last longer are high. And we are trying to understand why. And probably it's going to be different for every case, but yes, one of the things that are really important in humans and in mammals is the emotional part of a memory. If you, let's say, I'm sure you can remember, uh, or you cannot remember actually what you were having for lunch, let's say 2 years ago. It would be really weird if you can't remember. But if something else was happening at that time, like for example, there was an accident, or there was like the 9/11, I mean, you're probably too young, but something else happening in the world that was really important and it caused a really strong emotion in you, then you can remember all the other things that happened around that time very well. So you can remember how you had for lunch that day. You can remember what you were wearing that day. And so it's really clear now that emotions, strong emotions, are somehow enhance how we remember things. Let me see another question here.
I don't want to cut anyone off, but it is 10:52. We're a little bit behind schedule, so maybe
Okay.
just wrap up one more thing.
Okay, thank you everybody for listening.
Yeah. Okay. Thank you so much, Dr. Sandoval, for joining us. I hope everyone really enjoyed learning about the science behind, encoding and the consolidation of our memory.